Hello everyone, I am back with part 4 of animal cell culture and applications. Hope you would have viewed part 1, part 2 and part 3 of this particular chapter that I have uploaded in my YouTube channel. And I hope you have surely benefited from it. Key areas covered in this video: applications of animal cell culture, production of proteins through animal cell culture, and hybridoma technology. Applications of animal cell culture. Medically important protein pharmaceuticals are used in Chinese hamster ovary animal cell lines, and it is produced using a combination of two techniques: animal cell culture and recombinant DNA technology. So let's see what are these proteins. Number 1, erythropoietin, which is used against anemia. Anemia is a disease where there is lack of hemoglobin. Factor 8, which is used against hemophilia A. Factor 9, which is used against hemophilia B. Hemophilia is also called royal disease, and here the patients die of profuse bleeding as the blood doesn't clot. Number 4, tissue plasminogen activator or TPA, which is used against stroke. Number 5, follicle stimulating hormone, which is used against infertility. Number 6, interleukin 2, which is used against cancer. Number 7, human growth hormone, which is used against growth hormone deficiency. And finally, number 8, monoclonal antibodies, which is used for cancer therapy and autoimmune diseases. Coming to erythropoietin or EPO, it's a glycoprotein. It is involved in RBC production and wound healing. It stimulates the production of RBC and increases the oxygen carrying capacity of the blood. It is produced in response to two conditions: 1, oxygen shortage or hypoxia, and lack of oxygen or anoxia. It is advantageous over blood transfusion for the following reasons. Number 1, it do not require blood donors or transfusion facilities. And number 2, there is no risk of transfusion associated diseases. Factor 8. It's again a glycoprotein which is used for blood clotting, and in hemophilia A patients lack this factor. Factor 9, it is used to treat hemophilia B or Christmas disease. Tissue plasminogen activator. It's a serine protease. It catalyzes the conversion of plasminogen to plasmin, which is responsible for dissolving blood clots. So plasminogen, which is inactive zymogen, it gets converted to plasmin. Plasmin gets converted to fibrin, which is present in the clot, and TPA degrades this clot. It is used in patients having heart attack or stroke, and it is the first drug which is produced through mammalian cell culture. Let's see how these proteins are made. You will take a plasmid, which will act as the vector, and then the insert will be TPA gene insert in this case. Gene insert can be any protein of interest gene. This rDNA construct will be introduced into the mammalian cells. Mammalian cells, when it is overlaid with this recombinant DNA, will take up this rDNA, and this gets integrated into the mammalian genome. Then you select the mammalian cell secreting the TPA and you scale it up in a bioreactor or fermenter. Hybridoma technology for monoclonal antibody production, which is a very important topic. What is monoclonal antibody? It's a single type of antibody targeted against a specific epitope, and it is produced by hybridoma cell line. Cesar Milstein and George Kohler developed this hybridoma technology. They specifically bind to an epitope which is present on the surface of the antigen or an antigen itself, and it is used in detecting these specific antigens. They are produced by the fusion of two parental cell lines, one antigen activated B lymphocytes and two myeloma cells, which are cancerous and immortal. The fusogen is polyethylene glycol. So the hybridoma cells, which is the resultant of two parental cells, retain the property of both the parents. It retains the ability of the cells to secrete antibody and the ability of the myeloma cells to grow indefinitely. Let's see the difference between polyclonal and monoclonal antibodies. Polyclonal antibodies are derived from different B lymphocyte cell lines, whereas monoclonal is derived from a single B cell clone. In polyclonal, batch-to-batch variation affecting antibody reactivity and titer occurs, whereas monoclonal antibodies offer reproducible, predictable, and potentially inexhaustible supply of antibodies with exquisite specificity. Polyclonal is not a powerful tool for clinical diagnostic tests, whereas monoclonal, it enables the development of secure immunoassay system. Production of monoclonal antibody. Step 1. Immunization of the mice and selection of mouse donor for generation of hybridoma cells. Antigen, which could be an intact cell or whole cell membrane or microorganisms, will be mixed with the adjuvant for emulsification. This antigen will be injected into the body of the mice, and then spleen cells will be removed, which will be the source of B cells. Step 2. Fusion of myeloma cells with immune spleen cells and selection of hybridoma cells. Spleen cells obtained from the mice will be fused with the myeloma cells. Polyethylene glycol will act as the fusogen. The hybridoma cells, which is produced by the fusion, will be grown in a selective medium called HAT. B cells are HGR-PRT positive, Ig positive, and TK positive. Myeloma cells are HGPRT negative, Ig negative, and TK negative. Hence, hybridoma cells, which are a fusion, will be HGPRT positive, Ig positive, and TK positive. HGPRT is an enzyme which helps in purine metabolism. It converts hypoxanthine to inosine monophosphate, adenine monophosphate to adenine, and xanthine monophosphate to xanthine. Two important stages of cell selection. First one is the isolation of hybrid cells from parental cells. And number two is the selection of antibody secreting cells within the hybrid cell population. HAT medium, hypoxanthine, aminopterin, thymidine medium is used for the preparation of monoclonal antibodies. Aminopterin in the medium blocks the de novo pathway, and this medium allows the selection and growth of hybridomas which are HGPRT positive and have a normal functioning of salvage pathway. Applications. It's used in detection assays, purification techniques, gene identification, disease therapy, identification of cell surface markers, drug delivery, and dissolving blood clots. Most of its application is in the diagnostics and treatment. In diagnostics, it helps in early detection of many infectious diseases, whereas in treatment, it is used for the treatment of patients with leukemia, which is blood cancer, or transplantation rejection. Therapeutic monoclonal antibody OKT3. It is monoclonal antibody CD3, which is an immunosuppressant drug. It is used to reverse acute rejection of transplanted organs such as kidney, heart, liver, etc. It is the first monoclonal antibody which is used for the treatment of patients. And how does it work? T cells are the first army of soldiers which march against a foreign invasion. On its surface, it has a cell surface molecule called CD3. OKT3 goes and binds to the CD3 and block it. This results in the block of the function of T cells, and it will not cause transplantation rejection. So here is a picture of a liver donor and then there is a liver recipient. So you take the organ liver, then you extract the T lymphocytes and then you add OKT3 and then you put it back into the body of the recipient along with the organ so that there is no transplantation rejection. Herceptin or trastuzumab. This is approved for the therapy of early stage breast cancer, that is HER positive. These human epidermal growth factor receptors receive signals that help cells to grow and multiply. So what Herceptin does, it blocks these receptors. So the receptors cannot receive the growth signals, as a result of which cells cease to divide. And this impairs the breast cancer because in cancer cells divide indefinitely. So when the growth signals are prevented, cells do not divide and the breast cancer is impaired. Thank you so much for patient watching this video. I hope you would have got benefited from it. If you like it, please do click the like button. And if you have not subscribed for my channel for more videos in different topics of biotechnology, please do subscribe it. Thank you so much.